Good morning. So this is the other topic for today. Major histocompatibility complex. Right. So MHC or major histocompatibility complex is actually a large genetic complex with multiple loci. So for this presentation, you will be encountering some terms which are uh, used in genetics. So these genes that are in the AMHC are first identified as important in the rejection of transplanted tissue. So that's why the use of the word histo, that means tissue, compatibility was used. So another characteristic of this MHC is that they are polymorphic. So an example of polymorphism is when for example, the ABO blood group system, a person may belong to any of the four different blood types. You may be a type A person, B, AB, or type O. So one blood type, one blood group system, but you may have belonged to any four. Next. So in an experiment, they did an inbreeding of mice, and they show that the genes within the MHC are also involved in the control of humoral, that means antibody production and cell-mediated immune response. So additionally, I added this information. So this is also known as HLA complex or human leukocyte antigen. Right, so it's because of the structure that is present on the surface of the leukocytes. It's because most of the time we see this in the leukocyte um, surface. Right, so there are three classes. There are three classes. So the first one is class 1. Oh, by the way, it's found in the sixth chromosomes. The information about this MHC is encoded in chromosome number 6. So the first one is class 1 molecules. They are designated by letters HLA, B, C. It's not limited to these letters. There are others. And then for class 2 molecules, they are DP, DQ, DR, and so on. There are many others. The third type of MHC molecules is class 3. And this is related to some components of the complement and certain substances, cytokines like TNF-A alpha and TNF-beta. Right, so the function of class 1 and 2 antigens, this is from Henry, if you want to check that out. The function of these class 1 and 2 antigens are primarily in antigen presentation to T cells. We have seen this in the previous presentation. So these antigens are first discovered and their highly polymorphic nature was described in different research studies. And most importantly, they were found to be related to rejection of transplanted organs, human organs. Consequently, one of the most important clinical uses of this would be in screening for organ donation or tissue donation. Right, so another clinical application of this is that they are associated with some diseases like ankylosing spondylitis with particular class, which is HLA B27. Right, so another one would be the matching of platelets. There are some cases when platelet transfusions are not successful because of antibody formation after a person got received many units of platelets. So next. So we already mentioned this. There are three classes. And the products have many different roles in the recognition of self and non-self. It participates in the development of both the humoral and cell-mediated immune response. We already mentioned this, right? So antibodies may react with antigens alone, and T cells can recognize the antigen only when it is combined with an MHC molecule. So that is why this is very important. Without an MHC molecule, the T cell may not be able to see or recognize the antigen. The peptide that is associated with the given MHC, um, otherwise, there will be no immune response. Class 1 genes encode for glycoproteins, so mainly glycoproteins, and they are found in the surface of almost all nucleated cells. Okay, the concentration of course may vary, and the class 1 products are presented to the antigens 2 TC or cytotoxic cells, T cells. So they are found in the subloci designated as A, B, and C in the genes. On the other hand, for the class 2 genes, they also are mainly glycoproteins, and they are also expressed in the surface of the antigen presenting cells, similar to this one, right? So the antigen presenting cells are the macrophages, dendritic cells, B cells. We have mentioned this in the previous presentation, but they present these antigenic peptides to TH cells compared to the other one. They present it to the cytotoxic cells. So TH means T helper cells. Class 2 antigens are produced by these genes, loci in the genes, TP, DQ, and DR. And then thirdly, the class 3 genes encode for different proteins that participate in activation of the complement, okay, and the molecules that are involved in the inflammation. Right, so we can see here the structure of a class 1 molecule. So these two lines represent the cell membrane, and inside, this is inside the cell, this is outside the cell. So you can see that they extend towards the outside part of the cell. The composition of a class 1 molecule or chain is made up of two chains, a long alpha chain and a short beta chain. The beta chain is made up of beta 2 microglobulin. So the alpha chain itself is made of three different chains, alpha 2, alpha 1, alpha 3. So they are connected to each other. The four regions are cytoplasmic region that contains the site for phosphorylation. So if it's for phosphorylation, that is primarily to exchange energy or produce energy. Transmembrane region that contains hydrophobic amino acids. So in between, transmembrane region. So they do not allow the transfer of water molecule. Okay, and then what else? A highly conserved A3 region. So this is the A3 region which is able to bind with CD8. So CD8 is a type of TH cell, the helper cell. A highly polymorphic peptide bound binding region that is between A2, and A1. So in between this area is a site for um, allo antigenic binding. And then the B2 microglobulin is the one that's responsible for stabilization of the structure. Okay, so this one is the structure of the class 2 MHC. So you can see here that there are two polypeptide chains. So first this one, the alpha chain, which is made up of two um, chains, A1, alpha 1, and alpha 2. And then the other side we have beta 1 and beta 2. Okay, and you can notice that there is a carbohydrate attached to the surface of these polypeptide molecules. So again, the four regions are characterized by sites for phosphorylation. Okay, that is to produce energy and bind to the cytoskeletal elements. So again, they are embedded within the cell membrane up to the cytoplasm, and there is a part that is exposed outside. What else? You also find a region, the transmembrane region. So this area is hydrophobic, made up of amino acids. Okay, the alpha 2 and the highly conserved beta 2 serves to bind with CD4 cells. Okay, so going back to that CD8, that this one is CD4. Okay, a highly polymorphic peptide binding region. So alpha 1 and beta 1 region is the one that serves for binding for other components, other types of cells. So this is a comparison of the two types of MHC, 1 and 2. So again, they are embedded in the cell membrane. Look at that. And they are made up of certain regions, four regions. Okay, and this is an area that serves for binding to peptides. Right, so this is the designated locus or areas in the genes that corresponds to the MHC or the HLA. So there is the locus A, locus B, locus C. That is for the class 1 molecules, and then this is an additional class 1 molecules E, F, and G. But D is for class 2. So you can see that there are so many alleles that are part of these, form part of this locus. Okay, 118, 430, 96, and then for this one is only a few. That is why it's not the major locus for class 1 molecules. So again, they are expressed in nucleated cells, but the level of expression is variable. So that means not the same for all class 1 molecules. Okay, so the highest class is found in the lymphocytes, but they may also be found in fibroblasts. So they are responsible for wound healing in muscle cells, liver hepatocytes, and neural cells. So that means the nerve cells, but they serve as very low class of class 1 molecules. So this may be the reason why there are many successful liver transplants because of the low level of this molecule in the liver cells. So there is a less chance of rejection. In normal healthy cells, these molecules usually display the self-peptides. Okay, and then however, if they are infected by virus, the peptides from the virus will also show itself in the molecule. Unlike class 1 molecules, class 2 molecules are only expressed in the antigen-presenting cells. We already mentioned this, the macrophages, dendritic cells, B cells, and this is something new, thymic epithelial cells. So they are the cells that express these molecules. There is some sort of differences in expression, and it also depends on the maturation and degree of antigenic stimulation. So this is a representation of where we can find the class 1 and class 2 molecules. So they are found in nucleated cells, and class 2 molecules are mainly in the antigen presenting cells. RBCs? No, none of the molecules. That is why we call them human leukocyte antigens. So they are found in the WBCs. Again, so you will notice that we keep on repeating the same information. So they are found in nucleated cells. They are expressed in antigen-presenting cells for class II molecules. T cells that can recognize the peptides in the class II molecules are the T helper cells, while for class 1 it's cytotoxic cells or TC cells. Okay, so the expression of these molecules is regulated by cytokines. So this is again another role of the cytokines, that is to control the appearance of these molecules. So interferons, okay, they are related to viruses, and tumor necrosis factors related to class 1 molecules. Interferon gamma, okay, is also related to MHC genes type 1, class 1. Okay, so why? This is an example. They appear to induce the formation of specific transcription factors. So it has something to do with the transformation of RNA to DNA. So molecules can express self-antigens, and MHC partly determines the ability of an individual's T cells to respond to antigens of infectious organisms. So therefore it has been implicated in the susceptibility of the disease and in the development of autoimmunity. So sometimes they fail to recognize antigens belonging to microbes, and sometimes they also fail to recognize the self from non-self, leading to autoimmune diseases. So there are also other cytokines that can help in the expression of this molecule. For example, interleukin-4, what do? They allow class II molecules in the resting B cells. Okay, another one, B cells can also have these class II molecules, okay, regulated by interferon gamma, down-regulated by interferon gamma. And another one is expression of okay, the expression of this molecule is decreased in the presence of certain infections like CMV, hepatitis B, and adenovirus type 12. The responsiveness of this molecule, class 2 molecule, reflects the importance of these two T helper cells. Imagine if the molecules will not present itself, so therefore the TH cells will not be able to recognize these antigens. Okay, so that's how it is. And then it may result in the absence of immune responsiveness, as I said, and it may account for a relationship between the MHC haplotype and immune responsiveness to exogenous antigens. Some HLA alleles can occur at a much higher frequency. So these alleles can lead to the formation of certain diseases like autoimmune diseases. Maybe you have known some autoimmune diseases, viral diseases, disorders of the complement system, neurologic disorders, and some forms of allergies. So that is the relationship between MHC and disease susceptibility. To be susceptible means to be prone to developing certain diseases. So class 1 alleles are in this equilibrium with the class 2 alleles, and this can lead to, again, susceptibility to diseases. The differences in the alleles may yield differences in immune responsiveness. So in this case, the ability to present the process antigen in the antigen presenting cells is also affected. So it will be able to affect the ability of the T cells to recognize those antigen that were presented. And these allelic forms of the genes may encode for molecules that recognize as receptors by viruses or bacterial toxins. So the bacterial toxins and the viruses may uh, attach itself to the molecules produced by the MHC genes. Genetic analysis of diseases may show that some genes with multiple loci may be involved in complex interactions that lead to diseases. Although some individuals probably will not be able to develop an immune response, okay, to a certain pathogen, they will still be susceptible to infection. So the extreme polymorphism of the um, genes of the molecules will allow a species to respond and will be resistant. So that is the effect of this polymorphism. Some of the people will be able to develop resistance to infection. So in summary, those two molecules are very important. Okay, class 1 molecules are able to present themselves to CD8 T cells, and they note they are endogenous in nature, so they are already within the cells. So this is responsible for identifying viral cells or bacterial cells that have already been processed by these molecules. Class 2 molecules are those that present exogenous antigen to T helper cells or CD4 cells. So they are found in, again, in nucleated cells for class 1 and found in antigen presenting cells like B cells, macrophages, dendritic cells, and thymic cells. Right, so that would be the uh, important information. So they also encode for molecules that play no role in actual antigen presentation, class 3 molecules. Right, so some studies have shown that the haplotype, so half of the genes, can influence the responsiveness or the ability to present the antigen. Another one is susceptibility to certain diseases are linked to these alleles, certain alleles. Okay, so that would be the second presentation for today, the major histocompatibility complex. Okay.